Continuing on to another scientific talk, I would like to invite Dr. Rafael Pulido for giving a talk on characterization and functional reconstitution of CTNNB1 variants and its therapeutic interventions and implications. The floor is yours.
Thank you very much. Do I have sound?
Yes.
Okay. Thank you very much for this invitation, and I'm also very honored to see you again and to share some of our very humble results compared with the previous highly sophisticated research-like experiments that we have seen. I'm going to show you something much more simplistic because our work, we are focusing a lot on the patients themselves, and we are doing a highly specialized work on the genotype, phenotype consequences that we can find with mutations in this syndrome. I just have put from the beginning the name of the different people in Bilbao that is working with this project, which we call Ibero CTNNB1, because it's a consortium of at least 4 or 5 labs funded by several agencies. And the work I'm going to mention today to you is mainly made by us, Caroline and Anna, in our institute in Biobizkaia Health Research Institute. But at the end, if I have a moment, I will mention some other results from other collaborators that are working with us. So going back to the question that was made here, I was going to make my point on that very question regarding the important region, the important function of beta-catenin in the adhesive or the control of cell junctions. I'm not going to show results on that because we are also making more of our experiments in the other aspect of the but because it has been this question, I wanted to reinforce that point that actually beta-catenin was discovered with this function. This was later discovered, but we know much more about all this gene transcription reprogramming than in the function that beta-catenin makes in the cells and junctions. So I think we have to dig in research in this very area because maybe it's going to be critical for this very disease. Okay, and I also wanted to make a point on the particular property of this disease that is a gene, CTNNB1, that creates 2 completely different diseases. Here we are talking about one disease, which is generated by a mutation in the germline of the child, that we all know, but this gene is highly mutated in cancer, in the cells that create the cancer, not in all the body of the people, but in the what we call a somatic mutation. And this is a completely different disease, has nothing to do with the disease we are talking here, but it touches the very same gene. So this is a gene very essential that if you touch in one way or in another way, you can cause 2 completely different diseases. And actually, how the genetic causes of these 2 diseases are completely different. In the case of our syndrome, most of the mutations create what we call loss of function, that the protein doesn't work well. And in cancer, it's the other way around. In the cancer, the mutation make the protein to work too well, and then this creates cancer. And this is also a point that I want to make because the critical regulatory point of regulatory aspect of beta-catenin involving this, what is called the destruction complex. Beta-catenin is a protein that is continuously created in a normal person, and it's continually degraded by this destruction complex. What happened in our disease is that the body doesn't create enough because it's lacking one allele, and then you don't have enough to work. What happened in cancer is the other way around. It's not distracted enough. So the mutations that create cancer make an excess of beta-catenin in the cell and an excess of signaling. And this is, I think it's very important to have this in mind for therapies and to understand the disease, this particular disease, because it's a gene that is touching different aspects. We have a very short mini review now this year in Trends in Cancer that you can have a look because it exemplifies very well this concept. And then I'm going to the point of the meeting, the syndrome, the patients, the families, and the variants. We are focus our work in patients from Spain and Portugal. And we have been studying the functional properties of the variants that are potentially expressed in the children with this disease in these patients. I will show you some other results with another variants, but I'm going to focus a bit at the beginning on this. We already presented last year part of these results, and we have already published the functional analysis of plenty of Spanish and Portuguese variants. We have completed this assay, and this analysis, and I'm not going to show you again results, just to summarize. Almost all the patients we have analyzed the variants present nonsense or frameshift mutations, causing proteins that potentially have a different stability in the cell, and they are mostly loss of function proteins. This is a general rule that we now can say for the disease. It was kind of known already, but we have now tested a large number of samples as to be completely sure that the disease courses with loss of function mutation as an haploinsufficient classic disease. What we saw from this initial analysis is that the potential variant that could be expressed in the patients, and I want to mention this potential because we don't know if these variants are really in the body of the children, but at least we are studying them to know and to understand. We were able to define like two groups depending on the stability of the protein. So meaning proteins that maybe are more abundant, proteins that are less abundant. And we have now make an extra analysis of that to map with more precision this aspect. This is here summarized. And what we have come to the conclusion is that all the truncation proteins, beta-catenin proteins that stop before this region between the armadillos 9 and 10 are highly unstable, and most of the ones that extend farther down are more stable. So this is it's an important aspect to have in mind, because this kind of information, without making experiments, can give you a prediction on a potential variant. If you have a stop codon here, you have to go a more stable variant than if you have a stop codon here, as a general rule. Now we have made this as a general rule, and I think it's useful to have in mind. And also, what we have made is to make more general rules regarding the potential variants found in patients. We have mapped at the beginning and at the end of the protein which are the regions which are, let's say, useful for function. Before we were measuring just how the protein exists in the cell. Here we are measuring how the protein goes to the nucleus and how the protein regulates gene transcription, the previous talk. And it's interesting, this protein. This was kind of already known in the field, but we are mapping with high precision, and we can say that you mainly cannot remove things here or here because you need almost all the protein to be working well. And this is also explained by the disease. The mutations that are found in this disease, in the syndrome, are all around the protein. They are not in very specific spots. They are distributing all the protein. It means that the protein needs to have a minimal size to work. This structure, these armadillo repeats are very, from a biological and structural point of view, are very interesting because make a protein that looks like an accordion that moves. Very interesting. But again, not only for gene transcription, but also to go into the nucleus, which is, let's say, one of the major properties of this protein, to go to the nucleus to regulate transcription. You need basically most of the protein. You see that if you remove here things or here, then the protein doesn't go to the nucleus anymore. So we have made like a general view of the most of the mutations or the variants found in the disease, in the syndrome. But we were also very interested on another set of variants that have been defined for the patients with syndrome, with this syndrome. I think we have to invent an abbreviation for CTNNB1 syndrome, because I cannot say all the time CTNNB1 syndrome. We should call it C syndrome or something. I say syndrome because then there is in the literature and in some reports, there are described a panel of missense variants, missense meaning the protein exists, the patients have one wild type allele, and one allele that has one amino acid different in that protein. You know, the proteins have amino acids, and here there is these variants only changing one amino acid in the child. We have been testing already a wide panel of these variants, which are less abundant in the, let's say, history of the disease. They are less reported, but they exist. And it's very exciting, interesting, or frustrating, I don't know how to say, that we cannot detect alterations in the function of these variants, in most of them. We can detect alteration in some of them, which are especially clustered in the middle of the armadillo domains, which makes a lot of sense, which actually cause unstable proteins. This is, again, the very same idea with the haploinsufficiency. We detect missense variants that lose the function, but because they are not stable, and this is like they are not there, they get degraded. But most of them, by both methods, measuring the transcription or measuring the entry into the nucleus, they behave as the wild type. This is a very, I think, an important question to have in mind in the disease, that there could be a group of patients with what the geneticians call likely pathogenic variant that needs to be taken into consideration this likeliness. Are they really the pathogenic variant causing the disease? This needs to be proved. The disease already proves that there is a disease, but it's possible that there are another alteration that we are not detecting. It's also possible that here we are not measuring the appropriate function that is altered in beta-catenin in these patients. This is also true. We are measuring just 2 or 3 things, and maybe we have to measure 10 different things. But as far as we know, most of the missense mutations that are reported in databases, papers, or people that come in contact with us, we don't detect changes compared with the wild type. This is, I think, an important take-home message that we have to, at least, to be cautious. We have analyzed in high detail what it was initially called the negative dominance of beta-catenin variants in this disease, because this is important in order to implement therapies, and it was also actually taken into consideration for the clinical trial that is already running. We have made a very, very extensive and dedicated study of the potential negative dominance of the variants of mainly of the patients from our cohort, Spain and Portugal. And here you have all this as a group of Spanish patients. This is a group of Portuguese patients. And we have extended to most of the variants we have. And we don't detect any dominant negative activity, which again fits with the idea that we are with a classical haploinsufficient disease. We have not detected either gain of function mutations with these variants or with missense variants. Missense variants also for us don't manifest any negative dominant effect on the wild type allele that the patient has, nor gain of function effect. With this, I'm very simplistic in the way that the patient lacks one active gene. They lack one active gene. Now we have to dig to see exactly how is the activity, but they lack one active gene. That's how is the disease. And this is all the experiments that we have made in vitro go into that line. And I think can help to the design, for the design of the therapies and to learn more about the disease. I'm going to finish just showing this preliminary work that we are doing already. All the work I have shown you before is made in vitro with recombinant material, not touching a single hair from a child, all in vitro. The only thing we need is that you tell us the variant and we work. Now we are starting to work with the cells from the patients. And here I'm showing you some interesting examples of organoids or how these cells grow in vitro in the culture. And I think this is very preliminary, and of course we have to we have to study more cases and more in detail. But it looks that when you compare the cells, this is a Spanish case actually with a nonsense mutation. These are two cases that come from Simon's Foundation, not Spanish or Portuguese. But what you can see is that the morphology and the growth of these cells in vitro is different than, let's say, a control case, which is here, like this is a control case, or here. This is obvious because we are working with cells from child that they don't grow well. So it's normal that the cells don't grow well. But this, we think this is very important to have this work in mind to test therapies or to go into the potential further knowledge on the disease. And we are doing, in collaboration with a biobank in Spain that is collecting samples from our patients. I encourage all the Spanish and Portuguese patients to donate samples to CiberEarth, and they already know, and they are doing. And I just will finish with these conclusions, just mainly to open the discussion. Most of the mutations in the syndrome are nonsense and frame shift and result in proteins which, if they exist in the patient, have low stability in general and no activity and no evidence of dominant negative or gain of function mutation. All the protein is required for the activity, as I mentioned before. You can remove only small portions. The missense variants defined for the disease, as far as we know, they are a bit obscure, most of them, and the ones that are not obscure are unstable. And as I told you, there is clear effects in the cell growth in vitro of cells from patients. We are now working with additional beta-catenin functions to study in vitro, binding to effectors like the catenins, even binding to the proteins from the destruction complex. And we have assays that we are monitoring that, and also more with more derived. And at least to open the discussion for potential therapeutic approaches, that just for people to talk, many of us already know about the gene therapy. This has been, we are experts on that. But the inhibitors of things that will help, drugs that will help to the patient, I think are going to be very beneficial, maybe in coordination with some other aspects. I think next talk will be about the GSK3 beta inhibitors. Also, inhibitors of this kinase will be important, or anything that inhibits the destruction complex will be helping. We are making some work with, but I haven't showed you today because we don't have clear and not new results on what we call translational read-through, which is a specific approach for patients with nonsense mutations. And then any therapy that stabilizes a stable missense variant will help, but for this, we have to know that this variant is unstable, so you need to test it. And any thing that affects the expression of activity of the downstream effectors, as it was mentioned before, also will be helpful. And this is the people that have been working, Caroline and Anna in our lab are the main players here. We are also doing with some other laboratories in Bilbao, in a university, at Chucarro Center, and Universidad de Deusto. And thank you very much. And of course, the association, I cannot forget our strong connection with Estíbaliz and the Asociación Española CTNNB1, of course. Thank you.